The title of the paper is Simulating 500 Million Years of Evolution with a Language Model. This research presents groundbreaking work in the field of protein modeling and evolutionary biology. The authors of the paper are as follows: Thomas Hayes, Roshan Rao, Halil Akin, Nicholas James Safroniev, Denise Oktay, Ziming Lin, Robert Verkel, Vincent Kwai Tran, Jonathan Deaton, Marius Wigert, Rohil Badkundri, Irham Shafkat, Jun Gong, Alexander Derry, Raul Santiago Molina, Neil Thomas, Yusuf Khan, Cheta Mishra, Carolyn Kim, Liam J. Barty, Patrick D. Shu, Tom Surku, Salvatore Candido, and Alexander Rives. More than 3 billion years of evolution have produced an image of biology encoded into the space of natural proteins. Here we show that language models trained on tokens generated by evolution can act as evolutionary simulators to generate functional proteins that are far away from known proteins. We present ESM3, a frontier multimodal generative language model that reasons over the sequence, structure, and function of proteins. ESM3 can follow complex prompts combining its modalities and is highly responsive to biological alignment. We have prompted ESM3 to generate fluorescent proteins with a chain of thought. Among the generations that we synthesized, we found a bright fluorescent protein at far distance with 58% identity from known fluorescent proteins. Similarly, distant natural fluorescent proteins are separated by over 500 million years of evolution. The proteins that exist today have developed into their present forms over the course of billions of years of natural evolution, passing through a vast evolutionary sieve. In parallel experiments conducted over geological time, nature creates random mutations and applies selection, filtering proteins by their myriad sequences, structures, and functions. As a result, the patterns in the proteins we observe reflect the action of the deep hidden variables of the biology that have shaped their evolution across time. Gene sequencing surveys of Earth's natural diversity are cataloging the sequences and structures of proteins, containing billions of sequences and hundreds of millions of structures that illuminate patterns of variation across life. A consensus is building that underlying these sequences is a fundamental language of protein biology that can be understood using large language models. ESM3 stands out as a multimodal generative language model that simultaneously processes and generates data across 3 distinct modalities: sequence, structure, and function. The architecture operates primarily as a bidirectional transformer, which allows it to analyze context from all sides, both previous and subsequent tokens, when making predictions. ESM3 is trained using a generative masked language modeling objective, masking portions of input data and challenging the model to predict them based on other tokens. The largest variant, trained on a dataset of 2.78 billion proteins with 98 billion parameters, achieves remarkable representational fidelity for sequence, structure, and function. The ability to issue complex prompts to ESM3 opens up an entirely new dimension in protein engineering. By specifying a combination of structural and functional requirements, researchers can guide the model to produce innovative protein designs tailored for specific applications. The authors emphasize that ESM3 operates as a sort of evolutionary simulator. While traditional evolutionary processes are painstakingly slow, requiring millions of years for beneficial mutations to arise, ESM3 can generate functional proteins quickly, allowing exploration of distant expanses of protein sequence space. Today, we embark on a fascinating journey through the world of evolutionary biology and artificial intelligence. We're delving into groundbreaking research that showcases how advanced language models can simulate hundreds of millions of years of protein evolution, all while generating functional proteins that are remarkably distinct from known sequences. Protein language models, like ESM3, harness the power of natural language processing techniques and apply them to biological sequences. These models are trained on vast datasets comprised of protein sequences, allowing them to learn the complex relationships between amino acids, the building blocks of proteins. A key innovation in ESM3 is its multimodal capabilities. Unlike traditional models that focus solely on sequence data, ESM3 integrates not only the amino acid sequence, but also the 3-dimensional structure and predicted functions of proteins. This holistic approach allows it to reason across multiple dimensions of protein biology. The training process for ESM3 is incredibly complex and resource-intensive. The model was trained on a dataset containing 2.78 billion protein sequences, leading to a staggering 771 billion unique tokens. The training process involved data collection, masking strategy, and careful use of various techniques to enhance model performance. ESM3 stands out as a multimodal generative language model that simultaneously processes and generates data across three distinct modalities: sequence, structure, and function. The architecture of ESM3 operates primarily as a bidirectional transformer, which allows it to analyze context from all sides, both previous and subsequent tokens, when making predictions. ESM3 is trained using a generative masked language modeling objective. During training, a random subset of tokens across the different modalities are masked. The model learns to predict these masked tokens based on the remaining visible tokens from any of the input modalities. Now let's explore a significant application of ESM3, the generation of new green fluorescent proteins, or GFPs. The researchers prompted ESM3 with sequences and structures critical for chromophore formation. Among numerous designs produced, one standout was named ESM-GFP, a novel fluorescent protein with just 36% sequence identity to a well-studied GFP. This indicates an evolutionary divergence comparable to the evolutionary distance seen in nature over 500 million years. It is crucial to test the viability of these generated proteins. The authors conducted fluorescence assays and measured the brightness of candidates generated, confirming that ESM-GFP demonstrates comparable brightness to natural GFPs after maturation. Looking ahead, the authors suggest several avenues for further research. They advocate for ongoing refinement of language models to improve representations and predictive capabilities, including fine-tuning aligned with specific tasks to extract even more latent capabilities from larger models. In summary, the findings from this paper illustrate the transformative impact that language models like ESM3 could have on the fields of protein engineering and synthetic biology. These models represent a powerful new tool for generating novel proteins that may lead to advances in biotechnology. Figure 1a illustrates the iterative sampling process of ESM3. ESM3 begins with a sequence of masked tokens, representing an initial state for protein generation. At each time step, the model samples masked positions iteratively while considering context from previously generated tokens. This process maintains coherence in the generated sequences and facilitates realistic protein folding dynamics. Figure 1b presents an overview of the ESM3 architecture, highlighting the flow of information through input tracks and the processing pipeline. It emphasizes the integration of multiple modalities: sequence tokens, structural tokens, and functional keywords. Input tracks are converted into embeddings and fused into a single latent space for processing. The process of structure tokenization is depicted in Figure 1c. ESM3 analyzes local atomic structures to create discrete structure tokens. Frames are constructed for each residue, encoding both orientation and position in three-dimensional space. The output is a sequence of structure tokens that encapsulates essential geometric relationships, enabling efficient processing by ESM3. Figure 2a demonstrates ESM3's ability to follow prompts from each of its input tracks. The density of consistency to prompting is illustrated, showcasing strong fidelity and high foldability measured by PTM. Successful match to prompts indicates that ESM3 effectively integrates complex information across different modalities. Figure 2b illustrates ESM3's capacity to generate proteins that differ in structure and sequence from natural proteins. Prompt generations shift towards novel space compared to unconditional generations. This indicates ESM3's ability to innovate beyond its training set, generating proteins with significantly lower sequence identities. Figure 2c showcases ESM3's ability to generate creative solutions to various combinations of complex prompts. It reveals that the model can integrate prompts specifying functional sites and scaffold architectures. Many motifs do not show significant structural similarity to known proteins, illustrating ESM3's innovative generation process. Figure 2d presents an example where ESM3 compresses a serine protease while retaining the structure of its active site. This example highlights the model's ability to maintain active site coordination despite a substantial reduction in sequence length. Figure 3a displays the effect of fine-tuning ESM3 models on atomic coordination prompts. The tuning dramatically increases the fraction of tasks solved, showcasing the latent capabilities of larger models. This data indicates that preference tuning enables higher quality generations, significantly amplifying model performance at scale. Figure 4a illustrates the process of generating new fluorescent proteins, specifically how ESM3 is prompted with sequences and structures. Key residues for chromophore formation are highlighted, showcasing the systematic approach used to create new GFP variants. Figure 4b contains photographs of E. coli plates expressing novel GFP designs with quantification of fluorescence intensity. ESM3 successfully identified a notable design with low sequence identity that demonstrated fluorescence. This figure shows the fluorescence intensity of ESM-GFP over different maturation time points compared to natural GFPs. Although it takes longer to mature, ESM-GFP achieves brightness comparable to existing GFPs after sufficient time. Figure 4d presents analysis of the excitation and emission spectra of SMGFP. The results reveal distinct shifts in peak excitation compared to known GFPs, indicating its functional properties. In this figure, structural comparisons between ESM-GFP and its nearest neighbors highlight the mutations across the protein. The significant changes suggest the engineered nature of ESM-GFP and its functional robustness. Figure 4f displays the cumulative density of sequence identity across fluorescent proteins, emphasizing how ESM-GFP fits into this landscape. This comparison allows us to understand the evolutionary distance represented by the novel protein. This figure shows evolutionary distances against sequence identities among different anthozoan GFPs. It estimates that SMGFP indicates over 500 million years of evolutionary divergence from its closest known relatives. Figure 4h provides estimations of evolutionary distances by time, correlating them with sequence identity between different GFPs. This further supports the notion that successful designs can represent significant evolutionary timeframes in their divergence. Figure S1 illustrates the architecture of ESM3, presenting an overview that highlights the flow of information through the input tracks and processing pipeline. It emphasizes the integration of multiple modalities, sequence tokens, structural tokens, and functional keywords, which are essential for understanding complex protein relationships. Figure S2 depicts the geometric attention mechanism. Geometric attention enables the model to process 3D structural information, allowing ESM3 to reason about protein residues in a rotation and translation invariant manner. This enhances the model's ability to account for spatial relationships among residues during attention computation. Figure S3 displays the reconstruction quality of the structure tokenizer after 2 stages of training. Comparison of LDDT and backbone RMSD scores illustrates significant improvements in structure accuracy from the Stage 1 to Stage 2 decoder training. The results confirm that the model achieves high-quality reconstructions across various datasets. In Figure S4, random samples of structure reconstructions from the tokenizer are visualized. Most structures exhibit near-perfect backbone reconstruction, confirming the effectiveness of the training processes. However, the figure also presents some instances of poor reconstruction, often correlating with disordered regions. This figure showcases local neighborhoods that map to the same learned structure token. Rows represent different token indices, demonstrating how multiple local configurations can correspond to a single token. This highlights the model's decoding capabilities and the semantic coherence within the learned embeddings. Figure S6 assesses the calibration of predicted PLDDT and PTM scores. The results indicate that while most predictions are well calibrated, there is a slight overconfidence bias noted in some synthetic sequences. This reveals the need for careful interpretation of the predicted confidence scores. Figure S7 illustrates the schematic of function tokenization, detailing the process used to generate function tokens from protein annotations. Functionality is encoded using TF-IDF representations, which are captured and reduced to a manageable set of tokens. This method enhances the model's ability to predict functional characteristics when generating proteins. This figure presents the results from function prediction benchmarking. The mean average precision showcases ESM3's capability to accurately localize functional attributes within proteins. The model performs effectively across all keywords, especially notable for the top 1,000 prevalent keywords. Figure S9 visualizes the noise schedules applied during the training of ESM3. The probability density functions show how a mixture distribution is employed to balance representation learning and generative performance. This careful design drives the model's ability to predict and generate protein sequences accurately. This figure exhibits scaling curves for structure prediction, detailing how performance improves with model size and training data. The results exhibit that larger models significantly enhance predictive accuracy, especially on challenging datasets. Figure S11 shows the conditional and unconditional scaling behavior for each track in ESM3. It demonstrates that conditioning effectively improves performance across various tracks, highlighting the benefits of multimodal inputs. In Figure S12, the distribution of PTM and PLDT scores for natural versus generated sequences is compared. The results indicate a lower correlation for generated sequences, emphasizing the importance of ongoing refinement of generated proteins. This figure showcases the unconditional generation of high-quality proteins using ESM3. It visualizes the distribution of sequence lengths in the generation dataset and compares PLDT and PTM scores against the ESM2 model. Figure S14 illustrates the generation of sequences using a chain-of-thought approach. The comparison reveals that chain-of-thought generation yields higher confidence results and better structural outputs than direct generation. In Figure S15, ESM3 demonstrates its ability to generalize beyond its training distribution by designing proteins using SS8 and SASA prompts. The generated proteins closely follow the prompts while exhibiting low structural similarity to training set proteins, confirming the model's innovative potential. This figure highlights multimodal protein editing with ESM3, showcasing its ability to expose buried helices while conserving the original fold of the protein. This underscores ESM3's versatility in following complex design specifications. Figure S17 presents data demonstrating how model alignment improves the quality of generations. The distributions of PTM and CRMSD for base and aligned models illustrate that alignment enhances both fidelity and overall quality of generated proteins. In this figure, randomly selected successful generations from both base and fine-tuned models are displayed. This comparison shows the increased diversity and quality of ESM3's design outputs post-alignment. Figure S19 provides flow cytometry data, confirming the effective detection of ESM-GFP at a single-cell level. This validates the success of the designed fluorescent protein, indicating its functional viability in a biological context. This figure showcases the replication of design B8, indicating consistent results across multiple tests. The observation of similar fluorescence activity reinforces the reliability of ESM3's predictions. In Figure S21, the effects of chromophore knockout mutations on fluorescence activity are assessed. The findings confirm that specific residue mutations drastically affect the functionality of both B8 and ESM-GFP. This figure displays the sequence identity of ESM-GFP relative to various natural and designed fluorescent proteins. The findings highlight the evolutionary distance of ESM-GFP from known variants, emphasizing its novelty. Lastly, Figure S23 summarizes the evaluations of ESM3 Open, illustrating competitive performance in structure prediction and function prediction. The results confirm that the model maintains a strong ability to predict both structural and functional attributes effectively.